Can everyone hear me? Good. Thanks a lot for coming, and thanks a lot, Bill and Victoria, for that handsome introduction. So, my talk today is about rocks to humans. How does it really mean to be alive? What does it actually mean to be alive? So, you already know I work as a researcher at the University of Southern California. Interestingly enough, I actually work in the Gerontology department, so I'm technically a biogerontologist. Aubrey de Grey talk was awesome this morning. But my girlfriend's sitting there in the crowd, and she told me to not start my talk this way, because I was going to say, check this out, and she said, you can't do this. So we're going to skip that part. Very interesting. 4 in 10 Americans believe that the Earth, the universe, is about 10,000 years old, give or take a few. Only 42%, actually, yeah, 42% of Americans believe the creation version of life. So at some point, some extraterrestrial being put their hand in the middle, and voila, we had humans. So I'm going to present some things today that might be slightly more controversial, because that's the topics that I cover in my class. So it started all with our good old buddy, primate Homo sapien, and they were sitting around one day trying to figure out answers to life, because that's what people do. And one goes to the other and like, yeah, that's pretty much it. You hunt, you gather, and that's life. But of course, this crowd knows that the answer to the ultimate question of life is 42. But there are bigger questions, or better question rather. And I wanted to outline those as what is the origin of the universe, of biological life, and human life. I'm not going to talk about all of them because each of them is a discipline in itself. My outline is basically I'm going to cover Big Bang, which is the origin of the universe. And then I'm going to skip a little bit and talk about evolution, which is the origins of man as we scientifically know it to have happened. And then we're going to talk about the main part of the talk, which is actually what happens in the middle, which is a little bit more controversial than the other two for some reason. So, without further ado, Chapter 1, Origins of the Universe. This is how it all began. There was nothing except that little dot in the middle. We have calculated that dot to be the size of an end of a pencil. Nobody was there to see it at that point. But then, this happened. This is an artist's rendering of what the universe actually looks like today. They arrogantly put Earth and the solar system in the middle, which is probably not true. But at the same time, you are the center of the universe from where you stand at all times. So it's mostly true. To give you a little bit more detail, it all began 13.7 billion years ago with a Big Bang. At the beginning of that, there was no time, no space. And then you have the Big Bang and you have time and space. Inflation starts very quickly after. This is a matter of 10 to the negative 32 seconds after, is where you have electrons and quarks come into existence. These are the smallest pieces of life, or things that we actually know. Then, about 10 to the negative 6 seconds later, you're still under a second. Quarks aggregate and produce neutrons and protons. These are the nuclei of the atoms that we constantly interact with and are built of. A second passes, nothing is there yet. All we have is a super hot fog. At this time in the universe is so dense that light cannot pass through it. So we don't actually see anything. Everything is opaque. A little bit after that, about 380,000 years later, the universe is cooling down enough for electrons to slow down and allow them to be captured in the nucleus that we formed a little bit earlier, the protons and the neutrons. This is the very first time you actually have elements come to existence. Hydrogen, helium, lithium are the only ones that can actually come because they're the lighter ones. This also happens to be the oldest light in the universe. This is what we actually see when we look out in the deep space. We actually can physically see this. This is how we know, in one of the many ways, that how old the universe is. Nothing happens for 1.6 million years. This is during this time that the universe is cooling, gravitational forces are becoming strong enough to actually gather the dust and gases that are formed to form stars and galaxies. 900 million years after the formation of the first stars is where we have the first generation of stars to die. This is very important. If stars don't die, you don't have all the other elements that actually make life possible. Carbon, nitrogen, iron, and so on and so forth. So I wanted to give you a visual of what it looks like when a star explodes. It's actually very pretty. This is the Crab Nebula. This is first generation starts exploding. Nothing has happened yet. You need 3 generations of stars to die before you have enough material to actually make planets like Earth. So that takes another 9 billion years after the Big Bang. So now we have Earth, we have stuff on it, but there's nothing living. So we're going to fast forward, skipping the middle step, we're going to chapter 3. The origins of woman, man, human. I'm going to do this a little bit faster than the origins because I think this is more tangible for most people in this room at least. So it all starts with a gene pool. You need to have a gene pool to have some sort of a diversity. The gene pool can change based on the stresses in the environment. So you have your gene pool, and then things change. Food scarcity, new predators. Sometimes things come and grab one out. You need to be a faster fish to be able to escape that. Sometimes the environment changes. Now you need new ways to live in that new environment. All these are stressors that could potentially change your genome. Most commonly known as mutations. So what happens after that is you have to have a way to select for mutations that are beneficial and mutations that are not beneficial. The non-beneficials are easy, they just die out. The beneficial ones are the ones that have adaptive advantage over the ones that died out, and they survive. There's a lot of other things like symbiotic relationship, geographical locations, linear and nonlinear gene transfer, cross-species gene transfer, infection by viruses and bacteria, and so on and so forth. This is our current ancestry tree. This is everybody. It starts from the middle. And then, this is you. 99.99% of all living things on the planet have gone extinct. You are very small in compared to all of it. And here's the part that most people have a problem with evolution. Evolution doesn't say that you evolve from one thing to another and the other thing's still in existence. The new thing took the place of the old thing because it had an advantage over it. Here's a simple example for people to have a better understanding of it. Does anybody know of MRSA? Methicillin-resistant Staph aureus. It's a very deadly version of regular Staph aureus. It's antibiotic resistance, and it's really bad. We only have one cure for it called vancomycin, and we try to not prescribe that because if it gets resistant to that, we're pretty much screwed. So we stay away of it as much as possible. Interestingly enough, the regular Staph aureus is much stronger and more rapidly dividing than MRSA, except it doesn't have an advantage for antibiotics. So if you take MRSA and you take Staph aureus, put them in the same plate, within their cellular division, you will have a lot of staph, very few MRSA. Except we know how to kill staph, we just don't know how to kill MRSA. So advantage is just relative to their environment, not absolute. It doesn't make them stronger, it just gives them an advantage over something. To the main part of the talk. Louder. Better? All right, chapter 2, origins of life. The other big bang, or the other big show. So this is what it all starts with. I'm going to talk about inorganic molecules, rocks, organic molecules, we have to define those, and then protoviruses, protocells, and then cells and viruses. You have to get to the point of cells before we consider them quote-unquote alive. So, what is an inorganic molecule? I found this picture. It's gold, quartz, and iron oxide. It's from the Natural History Museum collection of minerals. It's quite beautiful. We consider that inorganic. So let's talk about crystals. What are crystals? Well, there's 4 different ways you can have crystals. There's covalent crystals like diamonds. There's a lot of people that like those. I find them overpriced. There's metallic crystals like silver, copper. And these are the things that usually have conductivity because the electrons are free around it. And then we have ionic crystals like table salt, sodium chloride. And then we have molecular crystals like sugar. These have different bonds, less strong as the other ones, but they still function very well. They're still crystals. So on that note, if you haven't already, make sure you check out the Natural History Museum because there are some crystals there that when you look at it, I highly doubt you would consider them not alive. They're fuzzy. They have fur. So what is the big difference between an inorganic and organic compound or a crystal? Well, crystals have encoded protocols. They can self-replicate, and they can autocatalyze their own self-replication. That sounds a lot like what living things do. Have you guys done this experiment before? A cup full of salt, a thread. You can actually hang something on the bottom to make it a little bit heavier with a pencil. Leave it around for a couple days if it's warm enough, and you have a crystal. It self-catalyzes its own structure. It has in it the codes necessary to make the crystal without any intervention. Well, what's an organic crystal? It is usually found in living beings, composed of carbon, hydrogen, oxygen, nitrogen. Those are the main components. Of course, it has many other things. And it kind of looks like this. Have you guys seen that organic before? It's sugar crystals. It's what you can sweeten your tea with. But most people think of crystals as the DNA molecule. This is an organic crystal molecule, right? The other one is too. We just use language in a different way when describing one versus the other. So what is the difference between those two? So on the left, I have here a topaz crystal, a blue topaz crystal. It's constructed of aluminum, silicon, oxygen, fluorine, and hydroxides. On the right, I have your DNA molecule with a crystallography image on the top. I mean, DNA molecule is a lot smaller, so you actually need X-ray crystallography to be able to see it. But the structure is actually quite similar. And it's made of adenine, guanine, cytosine, thymidine, phosphate, and deoxyribonucleases, or deoxyribosugars. And if you look at it, it's mostly carbon, hydrogen, nitrogen. Those bond in a very specific way. And when you put these things together, they are forced to only bind in the categories of things that they can bind to. They can't do something unique and new. It's always the same thing, which is why we can predict the shape of these molecules very easily. So the big question is, okay, sure, yes, DNA molecule could be considered inorganic if we don't call carbon things organic, and how do you go about making these things? Do we still need some sort of an extraterrestrial intervention to make this complex molecule? Well, in 1953, this is before the era where we had electronics, iPhones, internet, and all the cool things that we have today, they did this experiment, and apparently you can. You just put the elements in the primordial ooze of what we consider the young Earth, not the one that's 6,000 years old, the Earth that was actually young. And if you give it enough energy, which is in the form of lightning, which you had quite a bit of it back then, you can actually get your hydrogen, carbon, nitrogen, oxygen compounds, such as amino acids, ribonucleic acids, which is RNA, and DNA molecules. Which brings us to life. Life we know it today is defined under a certain dogma, which starts with DNA. That's where you have the coding information. Then that's protected inside a nucleus typically, or in case of bacteria, it's in a corner, away from most damage. That has to be transcribed into something that's communicatable. That's your RNA molecule. And in order to make those functional, you have to make it into protein. Well, that's cool. Let's see how we can make those, because if you can make those in a test tube, you have life, right? So the first thing you have to do is wrap it, or wrap it. That brings us to membranes. What do you wrap it with? Did I go too fast? Oh, sorry, animation issue. 1 minute? I got 10 on mine. It's a 25-minute talk. Yes. So we can have membranes made of lipids, we can have membranes made of proteins, or we can have membranes made of carbohydrates. You can also have membranes made of all these three things combined. In most eukaryotic organisms, you have membranes made of all three things. In simple organisms, you can have one, two, or other. I'm going to talk about cells before they become cells. These are membranes that those are lipid molecules inside water. So if you actually put olive oil inside water and shake it, it'll start bubbles. If you leave it to sit, they'll aggregate and create one bigger bubble. If you look at them under the microscope, they're able to create those structures without any change. You can influence it by adding, let's say, salt to the water, because that changes the pH and it will force the molecules to bind in a particular way. This is how you make vacuoles, which is basically the outside membrane of a cell. All right, we wrapped it. Now what? Well, once you wrap something, what does it look like? Let's talk about protoviruses. These are things that we don't necessarily consider viruses, but it has all the properties of a virus. It has an envelope, which we know how to make now, and it has some sort of a genetic material, which we also know how to make. So you imagine this, you're in the very early Earth, there's your primordial ooze, and in it you have lipids binding to each other, and you have RNA and DNA molecules happening on the side. This is all in a mix. Imagine in your jar and you can shake it, which happens naturally. Water moves and things get moved around. If you have an RNA molecule that happens to get wrapped into a membrane, all you need is one. Now the RNA molecule is protected from its environment. It doesn't get oxidized easily. And the lipids are covering that are considered a protovirus. Now, if the RNA somehow gives an advantage to that cell, it's the beginning of a virus. Just to go over what viruses actually are, these are some examples of viruses that we deal with today. Some of them has RNA as its genetic code, some have DNA as its genetic code, some have only membranes made out of protein, other have membranes made of lipids and proteins and carbs, and they cause some really good diseases. Panel A is a virus that infects bacteria, and the last one is the virus that infects bacteria. The two in the middle are the ones that we deal with. Okay, so I gave you all this talk, and there was a lot of thinking and imagining. Now let's see if you can actually see them. Because the big test for scientists is if we can actually reproduce this in a laboratory environment in today's world. These are man-made protocells. They're membrane-bound chemicals. There's no genetic code. There's no information other than the chemical properties of these compounds. On the left, you have one that interacts with its environment. It moves around to figure out what the density of the environment is. On the right, and we can play this again, on the right, it's the same molecule. When we add a substance in the middle, which is mostly sugar, it recognizes a source of energy and goes towards the center of that point where you have the most amount of sugar. Surprisingly enough, bacteria actually work around that way. They work towards the chemical gradient to find the more source of energy, and other than that, their movement is random. So we have actual live-looking protocells with no genetic material. Give you another example. Click. So the two left panels are two different protocells. They're either active or inactive, and depending on their activity level, they can interact with each other. This is socializing, or they can avoid each other. The one on the right is even more interesting. This is a little bit more complex. So you have about 10 different chemicals in those that makes the protocells. And they are able to interact with each other, engulf each other, and replicate. The protocells naturally, without any genetic material, figure out that their mass is too big, and then they divide. Let's go one more. This is protocells made out of primordial ooze. This is basically hundreds and thousands of chemicals now. Still no genetic material. On the left, we're looking at protocells just moving around, interacting with their environment, and even depositing things. On the right, we have protocells that react to a new source of energy. The droplet that fell was, again, sugar, and the protocells that are attracted to it, they interact with new sources of energy. Why is all this important? And how do we define the borders between life and no life? Well, it's easy to go from life to death, I think. Or is it? So a cell, as we define it within biology today, is the smallest unit of life. it has to be able to take energy, grow, and divide. I just showed you examples of things that can take energy, grow, and divide, yet by all definition, they're not living. So what I'm proposing today is actually life as we know it really simply does not exist. It's merely a continuum from rocks to humans. We just get more complex, and we have a more interaction between chemical bonds around us. And sure, in the way we figured out things to be more efficient, but other than that, we can draw a single line from rocks to humans. I'm out of time, so one last quick picture. These are prokaryotes, the simplest of the organisms that we know it. And these are eukaryotes, which we know came from prokaryotes eating other prokaryotes. One of them came out of existence about 1 billion years ago, the other one about 2 billion years ago. And humans, it took them 250,000 years by some accounts, and others 100,000 years to come to existence. So what I am going to leave you with is stay curious. Thank you very much. Oh, wait. All the animations was created by my very talented partner, sitting right there, who's a medical writer and another PhD. Now, thank you.
Yeah. All right. Questions in the audience? Questions?
Hi. How stable is the bubbles replicating?
As in timeline, how long they stay?
Yeah. Can you make it? I mean, does it scale up the way living organisms do?
So, yes and no. Those studies were done in Dr. Hankey's lab in Italy, and he actually has a TED Talk, which he explains this in much more detail and elegantly than I can. But depending on the nutrients provided in the environment, so a normal cell goes through the same requirements. You have to have energy coming in and have enough space for it to grow and divide. If you take any of those out, it just sits there. Ted?
How do you describe or account for the death of an otherwise living cell?
So,
as opposed to a cell that was never alive, and now a cell that is alive but then is dead. What's the difference?
My short answer to that is there's no difference. And I'll give you a little bit extended version. If you look at humans, when humans die, what happens? They stop eating, talking, interacting. That's what we can brain dead, and then biologically dead. They don't breathe. We consider them dead, right? But are all the cells in their body all dead at the same time? The answer is no. Most of them live for quite some time. And after, even when we consider all the human cells are dead, you still got the gut flora, which is full of bacteria. As a matter of fact, we have more gut flora bacteria than you have cells in your body. And they're still alive because that leads to the decomposition and moves on. So really, there's nothing. But a better answer would be that life starts with physics. Physics then creates chemistry. Chemistry creates biology. When biology reaches its end, it breaks down to chemistry again, and then chemistry breaks down to physics. It's very hard to say this is the time point where you're alive and this is the time point where you're dead. So what we do as humans, we create language to elaborate and have some sort of a definition, because otherwise it's very hard for us to talk.